Hey everyone, welcome back from our intermission. Thank you for tuning in to day three, the Psychedelics 2026. Up next, we have Alex Kwan joining us with their keynote, Psychedelic Drug Action, Cell Types and Circuits. Alex Kwan is a professor in the Meinig School of Biomedical Engineering at Cornell University. He's broadly interested in the neurobiology of drugs for treating mental illnesses. His research has revealed how ketamine and psychedelics act in the brain to enhance neuroplasticity. His lab is committed to open science by sharing data and code from its recent studies. Originally from Hong Kong, Alex received a B.A.Sc in engineering physics from Simon Fraser University and a Ph.D. in applied physics from Cornell University. Outside of the lab, Alex enjoys drinking a good cup of espresso and rock climbing with his kids. Alex, feel free to take it away from here.
Thank you for that introduction. Let me try to share my screen now. Okay. Um, Over here. Okay. Uh, Thank you for the introduction, and I'm glad to have this chance to tell you about some of the recent research in my lab. Most of my lab study various kinds of psychedelics, and particularly psilocybin. So on the left here, you see the chemical structure for psilocybin. And psilocybin and this active metabolite, psilocin, can be found in more than 200 species of these psilocybe mushroom, also known as magic mushrooms. Here you have probably one of the most famous species, Psilocybe mexicana. This one is quite well known because this is the one in the 1950s. Albert Hofmann was able to isolate psilocybin as the active ingredient. And then you have a couple other species with these beautiful photos. One grown in Chilliwack, BC. So this is near Vancouver, where I grew up and did my high school and undergrad studies. Then you have also one from Ithaca, New York, which is where I am at now, where my lab is. So these species can be found all over the world, and it really highlights the fact that these mushrooms, but also the psilocybin, have been used for quite a long time. And I would say, one side effect of doing this work is, I started collecting a lot of fungi mushroom photos. So if you have any good ones, do send it to me, and I really follow a lot of these on Instagram. And these are mushrooms and psilocybin are quite well known primarily because of their acute behavioral effects. As you all probably know, they induce subjective experience. Some of the best account of this, actually, in my opinion, came from this Danish study where they asked a female participant after administering one dose of psilocybin what they saw. So they drew that on the left, and then also how they felt. So this person wrote, I had the feeling that I was experiencing the world through myself as a little girl holding her dad's hand. My dad and I were observing what was happening around us. I think we saw something that resembled beautiful nature and charming castles. So you can really see the type of perceptual change and emotional change. Some people describe this as connectedness with the universe, with other people that happen. And for oral administration for psilocybin, these type of experience typically has an onset about 30 minutes to an hour and then lasts for anywhere from 4 to 6 hours. But what's exciting about psychedelics is the therapeutic potential. And this has, I think, really re-emerged over the last 10 years or so. And this is one of the probably latest published study, although now there are even bigger phase 3 clinical trials. But this is the one from Usona Institute, where they show that one dose of psilocybin with psychological support can lead to reduction of depressive symptoms. These are individuals with major depression. You can see in this phase 2 trial that within 8 days, you can see that big difference in the depression score. And what's most striking is that after one administration, you can see that long-lasting effect out to day 43. So this holds again in larger trial, and there's every indication to suggest that it would be the case given the recent Compass Pathway trial and also the LSD trial. Um, then I think this kind of research can and availability of the drug can really transform psychiatry, given the possibility of using these drugs as treatment. So as a result, I think there's a lot of hype in the field, both in terms of uh, you know thinking about these as therapies. So you have New York Times saying psychedelic revolution is coming. Psychiatry may never be the same. You have Newsweek saying that this is a new treatment for depression. And then you also have some other headline. For example, this one, elite golfers turning into mushrooms and um, and psychedelics to dominate the PGA Tour. So these are more fun and headline. But this one from economists, which I like a lot, economists always with great puns, business, the ultimate business trip, and bosses want to feed psychedelics to their staff. And then finally, the tagline, are they high? And then here, NPR say the National Park Service wants people to stop licking this toad. And obviously, they're referring to the Sonoran Desert toad in Arizona and New Mexico, which has 5-MeO-DMT. So I think one thing about doing this type of research is I think there's now a lot of media attention, which makes it very fun and impactful to do, I think, psychedelic science. But also, I think as a researcher in this area, if you also feel a certain amount of responsibility, I think we all share this responsibility to communicate effectively and also communicate the science accurately. So I think this is something that we should all strive to do, and definitely something that I'm quite mindful of. But today I'll spend the time telling you about the science behind psilocybin, where I think the field is and where the field is going, and give you some examples of that from my lab's own research. So we know quite well that psilocybin is what's known as a prodrug, where it metabolizes into psilocin. And psilocin is what crosses the blood-brain barrier, enter the brain, and then causes the behavioral effects. Many of the molecular target is quite well known. Psilocin is an agonist at the serotonin receptor, and there's a lot of effort to understand the range of serotonin receptor that it acts on. Here's some work, including quantification and measurements from a recent study from Brian Roth's lab, and also some known value for serotonin. I highlighted some of the receptor subtypes. You can see that 2A receptor is known as the key receptor. You can see that both psilocin and serotonin bind to that receptor. 2B also both bind quite strongly. This is the one that everybody want to engineer away because it causes cardiac trouble. And then interestingly, there's also the 5-HT3 receptor, which is the inotropic receptor that serotonin bind, but interestingly, psychedelics do not bind. But even though you have these small differences, you can see on the whole, a psychedelic like psilocin do bind to a pretty wide range of serotonin receptor, just like serotonin. But what might be different is what's shown here on the right. You can see that even though they bind to the same set of receptor, the relative preferences, we think there are differences which lead to some of these nuanced behavioral effects for these drugs. So understanding that would be quite interesting. Even though we know the target quite well, which are the serotonin receptors, I would argue that at the neuron level, where these receptors express, how the drug affects individual neurons, and also at the neural circuit level, how the drugs affect the neurons that are embedded in the different brain regions, is still at a very early stage, and we don't know what the drugs do. And understanding how psychedelic act on neurons and neural circuit, I think will help a lot in terms of accelerating drug discovery efforts. So for today's talk, first, I'll actually spend some time telling you about some of the early work that my lab has done. I think it really set the stage on what we're doing right now. After going through some of that early work, and then hopefully that will motivate you why we're asking the questions that we're asking today. Then I'll tell you a couple of studies that is ongoing in the lab right now, and then finally give you at the end a perspective on where I think the field may be going. So our research started in late 2010, like around 2019. And around that time, we have some fairly basic questions. I think the field in general, in terms of preclinical research, was still at quite an early stage, where the clinical side have restarted. But in terms of animal studies, it hasn't been done as much. These are just very basic questions. What doses should we be studying? Which brain region are affected by psychedelics? Can psilocybin induce structural plasticity? And the last question is quite specific, motivated at that time by a series of very elegant work from people like Ron Duman and Lisa Montega about how some other drugs like ketamine could induce structural neuroplasticity. So we were also very interested in whether psilocybin could also have effects on structures like the dendritic spines in neurons. So one of the very first study we did was this one by PhD student Clara Lau and Ian Gregg. Here, again, when we first got the drug, we didn't quite know what we have. Clara and Ian administered a single dose of psilocybin to mice and observed the very classic head twitch response. Here, the mouse on the left, you can see the head twitch right here. It's like a sideward head movement. We recorded with a high-speed camera and then slowed down so you can see the individual head movement. And you can compare it to the mouse on the right, which has received saline. And then we map dose response, which is quite important when you're studying a drug. What you can see is that at a dose of about 1 milligram per kilogram, you start to have that psychoactive effect where the mouse start to exhibit the behavioral effect of the head twitch response. So this is a dose that we'll use for many of our study that I'll tell you today. That's 1 milligram per kilogram, which we believe is a psychoactive dose for the mouse corresponding to the probably like a full psychedelic dose in human. We also look at some longer-term behavioral effects. So this was work done by postdoc Sam Woodburn. Not only do psychedelic have these acute effects like head twitch response in mice, but it can also have some longer-term effect like seen in clinical trials in human. So one of the standard assay in mice is fear conditioning. Here, Sam pair auditory cue with a foot shock in mice. Then later on, test the ability of a drug to let the animal extinguish that association. So what you can see is that after a single dose of psilocybin, if you immediately test extinction, you can see that the mouse already froze less. So here, in response to the auditory tone, they show less freezing experience, meaning that they were less responsive to that initial sort of traumatic, stressful experience. But what's more interesting is after the drug has gone out of the animal's brain, we continue to see this reduction in freezing behavior, suggesting that there are also long-term effects in animal models, maybe in parallel with some of the longer-lasting beneficial effects that people have seen in clinical trials. So all of this is to say that, I think this 1 milligram per kilogram dose, or within around this range, 1 to 2 milligram per kilogram dose, is probably a good range to study, both in terms of the psychoactive effect of the drug, but also some of the long-term behavioral changes. So then, we next want to figure out, study the brain, but where in the brain is it acting? Even some of this, again, simple question was not so clear at that time. So MD PhD student Pasha Davodian did a brain clearing study. So you can see on the top there, on the left is a normal mouse brain. It's very opaque. It scatters light. On the right is a clear brain. So he embedded this in hydrogel and basically clear out all the lipids so you can now see it is optically transparent. And using this method, he can image the expression of this immediate early gene CFOS in the brain, which relates to activity of neurons. And what he found is that after giving an animal a single dose of psilocybin or ketamine, a lot of brain regions have CFOS signals, suggesting they're responsive to the drug. So some of these regions are in the neocortex, like the anterior cingulate cortex. And I highlighted that because that's where we spend a lot of our effort on studying, but also other areas like somatosensory cortex, auditory cortex, visual cortex, such as spinal cortex, and subcortical area too, like locus coeruleus, lateral habenula, colostrum, basolateral amygdala, and central amygdala. These are common share region between psilocybin and ketamine, which is another rapid-acting antidepressant. But there are also some regions that's psilocybin specific, like the dorsal anterior insula, which is quite interesting because this area integrate the body's internal state and then drive motivated behavior. Periaqueductal gray, which is involved in sort of this fight or flight experience. And then finally, dorsal-raphe. So this, I think, was give us some hint on where the drug act. We actually went on and did a bit more work on this, mapping more drug and maybe thinking of this more of a drug development tool. This was work by another MD PhD student, Farid Abouhab, basically taking some of these CFOS map and asking AI, can you, based on these CFOS map, guess what drug was given to the animal? And what you see on the right here is that the AI can guess quite well between things like psilocybin and 5-MeO-DMT. It guessed correctly 99% of the time. Psilocybin, MDMA, 94% of the time. The time when the AI couldn't do actually very well was when we compare psilocybin with a non-hallucinogenic 2A agonist like 6-fluoro-DT, where it seems like, at least in the CFOS level, the psychedelic versus the non-hallucinogenic analog give about the same type of CFOS activation in the brain. So now that we know what dose to study and then what brain area we act on, we were very interested in whether the drug has any effects on synapses in the brain. So this was work that's spearheaded by postdoc Ling Shao. So she used a method called two-photon microscopy. This is in vivo imaging in a live mouse, the neuronal architecture in the brain. So if you come to my lab and look under the two-photon microscope, you might see something like this. Here, the neurons are labeled with fluorescent protein, and what you see is the dendrite that comes out from these neurons. And you can even see the little protrusion out of the dendrite, which are called the dendritic spines, which is where most of the excitatory synapses are on these pyramidal cells in the brain. What Ling Shao did is give the mouse a single dose of psilocybin and look at how those protrusions, the dendritic spine, changed. And what she found is that there's about 10% increase one day afterwards that persists actually. So when she looked at it 34 days later, about a month later, she can still see more of these dendritic spines in the brain. So really giving some evidence even in a living brain, a single dose of these drugs can have fairly long-lasting structural plasticity effect. It really remodels some of the neuronal connections in the brain. So this was already 5 years ago. Since then, several people in the lab have extended some of these studies. And we also did a literature review combining with other studies of drug effects in the field. What you show here is sort of combined result where there's obviously some minor experimental detail, but it gives you a hint on overview on what these drugs might do. You can see that the structural neuroplasticity effect is actually shared across a number of drugs, things like ketamine, 5-MeO-DMT, which was imaged by Sarah Jefferson, a psychiatry resident in the lab, or that first study on psilocybin. They both share this ability to increase the neuroplasticity. Even some other things does as well, like cocaine and allopregnanolone, which is a drug for postpartum depression. Some other drug like benzodiazepine, particularly diazepam, actually have the opposite effect where it reduces spine density. So I think it's quite interesting to see the timing of the change and also the magnitude of the change and compare it across the drug and see, it give us some hint on how the different drug might have different plastic effects on the brain. So at this point, we were quite excited about some of these findings. And then over the last couple of years, we decided to really dig deeper into this plastic changes and ask some mechanistic question about the features of the plasticity. For example, whether some of the serotonin receptors are needed. And then also, if we have a synapse, given that we have the postsynaptic dendritic spine, there must also be an axonal input on the other side. What are those? And those are the questions that we want to answer. So for the first question, Ling Shao teamed up with Ph.D. student Clara Lau in the lab using knockout mice to knock out the serotonin 2A receptors in pyramidal neurons quite sparsely. And what they found was a very striking result, which is if you do that, the psilocybin evoked structural plasticity is completely abolished. So what you see here is the yellow curve showing the spine growth that we see quite a number of times now when you give mice a single dose of psilocybin. But when you knock out the 2A receptor in these neurons, we really no longer see that spine growth. That thing is completely gone, even though the mouse receives psilocybin, suggesting that 2A receptor is essential for this process. We're also quite interested in what are the inputs going into these synapses. So this was work by another postdoc, Kwan, working with the Allen Institute. Here we use a different method, which I won't go into the detail except to show you a movie because it is quite involved. But if you're interested, you can look at this recent published paper. But we use a viral tracing method using the rabies virus. And the kind of movie that you get, the data looks something like this, where the red cells are the cell that we're tracing. Green cells are all the cell providing the axonal input. So here's going through the brain. You can see a lot of cell providing input, and here's the starter cell in red. We can compare brains that are from mice receiving saline or brain from mice that receive psilocybin and look at how the input cell, the number of green cell, changes from the different brain regions. On the right shows you a summary of this result, where we find that psilocybin strengthened input from some areas, like the retrosplenial cortex, visual cortex, and also some sensory motor cortex, but actually reduces input from other areas like the lateral network and the VMPFC. This work you really dig into the deep mechanism, and we have some idea that some of these input changes might be related to which area might be having more stronger neuroactivity. So Kwan went on to test some of these ideas first by recording at these different input regions, but also finally show that if you silence one of these regions, you can affect how the drug changes the remodeling. Again, just giving deeper insight into what is that plasticity process and what kind of inputs are we talking about? What circuit are we remodeling with psilocybin? So that's a very quick overview of actually 6 years of work in the lab. Showing you how we have this series of study really digging deep into how different psychedelics, particularly psilocybin, lead to long-lasting structural plasticity. Then we got quite interested, and rightfully so, to really want to know the underlying mechanism. So figuring out that the 2A receptor is essential for this process, but also mapping, yes, we have plasticity, but what specific circuits are being strengthened. Yeah, we were very excited about that work, but now I'll tell you about what we are doing now and what I think is really important for next step for understanding the drug action. Before that, I do want to acknowledge the lab. So this is the lab. Currently, we have 5 PhD students and 3 postdocs. Most of the lab is funded through NIH funding. So we have a lot of support from NIMH, the National Institute of Mental Health, and then also a small amount of funding from private foundations. So one of the things that I maybe talk about very briefly, but really didn't get into it, is that we find that psilocybin induces structural plasticity, and we focus a lot on the changes that happen on the pyramidal cell, which have these excitatory synapses. But the brain itself has many more cell types. Some of them are neuronal cell types, some of them are non-neuronal cell type. And it is really likely that the drug effect actually involved the broader circuitry. So not only these specific excitatory cell type, but likely many other cell type that actually exist within the brain. Some of these are GABAergic interneurons, so provide inhibition, silencing other cell types. Some of these are things like astrocytes, microglia, and oligodendrocyte. They're a non-neuronal cell that provide also critical function to support brain function, but they don't necessarily have synapses to communicate spiking action potential information. So we got quite interested how the drug might influence these other cell types. So this was one recent study now by PhD student Clara Liao with help from another PhD student, Amanda Weiner. What we did here is leverage new genome-wide sequencing study. So this is a method called single nucleus RNA sequencing, where Clara and Amanda took a number of mice, including control mice that have not received any drug, and then mouse that received psilocybin, but then were sacrificed 1 hour, 2 hour, 4 hour, at different time point after the administration, and then a parallel group that have been administered ketamine and then sacrificed at different time points. Clara and Amanda would microdissect the medial frontal area, which is where I've been measuring the spine changes, dissociated the cell, and then sequence the RNA transcript within that area to look at how the gene expression have changed after the drug administration at the different time points. So this was actually work in collaboration with a couple of collaborators from Michigan, Ethan O'Farrell, who did a lot of the computational work, as well as his PI, Ken Kwan. So with this large-scale data, what we did is apply Allen Institute's Map Myself pipeline to ask, what are each of the cells in our data set? How do they match on the known cell type within the frontal cortex? And as I show you, there are a lot of excitatory cell type. So all of these ones on there are 16 cell type. This is big data, but all of these ones, L6IT, L5IT, all of these ones on the left 2 columns are excitatory cell type. Then you have 4 inhibitory cell type, VIP, LAMP5, P-valve, and SST. Finally, you have non-neuronal cell type, astrocyte, OPC. That's an oligodendrocyte precursor cell, and then oligodendrocyte as well as microglia. What this show you is that we have about 61,000 cells for control sample over 100,000 cells each for psilocybin and ketamine, and we can really segregate and identify these cell types quite confidently in our data set. We can ask first, given that we're interested in psychedelic, where are the serotonin receptor in this cell type? Given that we know the drug binds the serotonin receptor, figuring out where the receptors are would be quite important because then we can know whether the drug can even bind to that cell type to exert its action. You have HTR1A, which expresses the gene encoding the serotonin 1A receptor, HTR2A for the 2A receptor, and 2C. What you can see is that for the 2A receptor, it expressed in a lot of excitatory cells. That's why you see that blue cloud in the middle, but actually less so in some of the other cell type that is characterized in our data set. 2C has quite a different pattern, particularly high in some of the interneuron data set subpopulations. And then the 1A is much sparser in this frontal area. We can quantify that differences. So this is what's known as a dot plot. Bigger the dot means more transcript, and then the deeper the color means more transcript. The more transcript means the abundance. How many transcript exists? The dot size means like how many of the cell actually have that transcript. But what you want to know basically is that bigger the dot is, again, more cell, and then deeper color, also more level of transcript. So you want a dot that's both deep in color and also big. On the top is the 8 excitatory cell class. Again, you can see that they express a range of serotonin receptor. Then you have the interneuron, which is also express some serotonin receptor. What's quite surprising to us is actually many of the neuronal cells don't really have a lot of transcript or serotonin receptor. You can see it's mostly blank on the bottom 4 rows. So we can look at what genes are changing after the drug is administered. So here showing you just one cell type. This is layer 5, these pyramidal tract neuron that we have been quite interested in. You can see that at 1 hour time point in this so-called volcano plot, there's quite a lot of gene that's changing. Some of them increase, upregulated, some of them decrease, downregulated, decrease one in blue, increase one in red. By 2 or 4 hour data point, the number of gene changes start to decrease. And then by 72 hours, quite interestingly, the number increase again, suggesting that they're both an early phase and then a late phase of gene expression transcriptional changes. We can not only look at just one cell type. Again, we have 16 cell type. The pattern largely hold. So here I'm showing you the 5 different time point with the 16 different cell type all in one plot. You can see that many excitatory cell type actually show transcriptional changes, also some of the interneuron type, even some of the non-neuronal cell, which we find quite interesting. So even though they don't have serotonin receptor, these non-neuronal cells certainly can respond to psychedelic, probably through indirect circuit mechanism. And you can also see that that initial wave of transcriptional response right at 1 hour, and then followed by a late phase at that 72 hours, which again is actually circuit-wide. So these really give some insight on how the molecular program within the brain change and also across different cell type in the frontal cortex. We can look at what these genes do. So in an excitatory cell, here's one example, GRIN2A, which is actually a critical subunit for the NMDA receptor. You can see that that gene increases early in a number of these excitatory cell type. And in general, when we do gene ontology analysis, you can find that many of these genes are related to synaptic transmission, glutamatergic synapses, regulation of neuron projection development. So really consistent with the idea that you're promoting structural plasticity, the synaptic level changes in these neurons that we have observed previously with our imaging methods. When we look at inhibitory neurons, the genes are actually quite different. Here's one example called KCNQ3, which is actually a potassium channel that regulates the excitability of the cell. We actually see a lot of more mitochondrial-related genes. So here, aerobic electron transport chain or cation transmembrane transport, regulating the excitability of the cell, ATP synthesis from the mitochondria that really help with the metabolic function of the cell. So the significance of this is not really well known. This is just what we observed, which was quite interesting and quite distinct from the excitatory cells. And finally, as I mentioned, non-neuronal cells also show quite a bit of transcriptional responses. Again, even though they don't have serotonin receptor per se, at least not a whole lot from the transcriptomic studies. And here show you oligodendrocyte and OPC actually have quite a lot of this change in this gene, which is NLGN3. Many of these genes related to neuron projection guidance or axon guidance or axonogenesis, again, maybe supporting some of those plasticity changes that's occurring in the excitatory cells. So it is, again, big data set. I can't show you everything. The idea for this was to develop a resource for the community for each of you, if you're interested, to go in and dig in and look for your gene of interest in the things you want to study and see where it's changing. So we've actually made this interactive web portal that anybody now can go in and you can type in your gene. You can even type in several genes, plot the traces that like the ones I just showed you, as well as download all the data for a few genes, all the genes. We both put in the raw data as well as the process data. So you can really play with this data set, which hopefully will be quite useful. And you can also compare with the ketamine, which I really didn't talk about. You can compare between the changes with the psilocybin, with the ketamine, which already in our early analysis showed some quite interesting changes in terms of the time course of the transcriptional responses. Now I switch to second project that I'll tell you about today that's ongoing in the lab. So one of the consequences of this type of analysis is that we also found that inhibitory neuron, which we have not studied so far, also express an interesting complement of the serotonin receptor. Unlike excitatory cell, the expression here seems to be quite cell type selective. So you see that the SST cell have some 1A expression, whereas the parvalbumin cell and LAMP5 have 2A expression, VIP cell mostly 2C expression. Again, unlike some of the excitatory cell, which could have all of these, 1A, 2A, and 2C within the same cell. So we were very curious what that means in terms of the drug effect on the interneurons. So MD PhD student Pasha Davodian did this experiment where he records now the spiking activity from these interneuron population. The way he does it is he would express channelrhodopsin for optogenetics in one of these interneuron population, for example, SST interneurons. He would record using NeuroPixels probe for 30 minutes, infuse the drug or saline vehicle into the animal, and then record after the drug was administered for another 60 minutes, comparing pre and post differences in the firing rate. And then finally, he would do what's known as an optotagging to identify the cell. So how it would look like is when he do the recording, this is kind of the raw traces. You can see that because these are high density probe, he record from many channels. Each of those little line is a spike in the trace, and some of the spikes can be captured across multiple sites on his probe. And then finally, he will do this optotagging, which is he would shine laser light onto the surface of the brain. And those neurons that express channelrhodopsin would elicit spiking firing activity. So he can figure out which one actually was the SST neuron among all the cells that he has recorded. When he does this, he found that the SST neuron seems to reduce their firing rate after administration of psilocybin. So here on the left is saline, pre-drug firing rate, post-drug firing rate. You can see that they lie mostly along the diagonal. But for psilocybin, most of the dots are below the diagonal, suggesting that they have reduced their firing rate after the drug was administered. And we can plot the firing rate over time, so you can also see how that changes over time. And indeed, you can see that decrease in firing activity in the SST cell. And then now he did the same thing for PV cell, and interestingly, it showed a very distinct response. So unlike the SST cell, here all the dots lie above the diagonal, which means that the PV neuron increased the spike rate after the drug was administered. One thing that Pasha noticed was that the changes in the firing rate was somewhat delayed. So you can see the changes in 30 minutes, 60 minutes around that time. So we were wondering how that corresponds to drug level in the brain. So this was done by a couple of students in the lab, Corey Knox and Amelia Gilbert, where they administered psilocybin to mouse and then extracted the brain tissue and basically run mass spec to look at what is the brain level of these drugs, of psilocin. And this is the data that they got, and we fit it to a one compartment model. You can see that the drug takes about 30 minutes to get into the brain to peak level, and then another half-life of about 56 minutes, about an hour to leave the brain. So we think that some of the spiking activity changes we see correspond quite well to this time course, where it takes again about 30 minutes to go in and then an hour to wash out from the brain. Again, figure out this firing activity changes was quite interesting, and the psilocybin and psilocin can cause us these distinct changes in the firing activity. But we also want to understand why that is the case. Why do we find that SST cell decrease activity, but the PD cell increase activity? There are a couple of possibilities. One is that these cells connect with each other. So this other class of neuron called VIP is known to inhibit the SST cell. It's possible that psilocybin or psilocin act on this and then suppress the SST cell. Another possibility is that, as I mentioned, SST cell express 1A receptor. So it's also possible that the drug just directly act on the SST neuron. So we want to figure out which or maybe both of these pathways may be contributing to what we are seeing so far. For the VIP neuron, student Neil Zavalia and Josh Wilson did some imaging study. These are calcium imaging study. Basically, what they found is that comparing pre and post-drug administration, really didn't see any effect on these VIP interneuron, suggesting that likely is not through this disynaptic VIP interneuron pathway that lead to the drug effect on the SST cell. This leaves the other possibility, which is maybe the drug act directly on that 1A receptor, reducing the firing rate. So we tested this by, again, creating knockout mice. So we will get this mouse line from a Japanese collaborator where we can knock out the 1A receptor selectively in the SST cell. We did some validation here using RNA-scope to show that indeed, when you cross the mouse, the transcript for the 1A receptor is now substantially, significantly reduced in the SST inner neurons. Although you noted that it's not zero, but it's substantially reduced nonetheless, from about 40% of the cell containing the transcript to now about 10%. Now, when we record it from these mice with the 1A receptor knockout, we find that psilocybin no longer reduced the firing rate, suggesting that that 1A receptor on the cell type is critical for meeting this suppressive action. So overall, to now conclude some of these ongoing study, we did actually one more study here that I should mention also, which is that we also take these 1A receptor knockout mice and run some behavior to show that it also has some behavioral relevance. When we run, for example, a depressive-like assay, tail suspension, you find that the SST1A knockout mouse actually no longer show the drug effect on immobility, suggesting that this interneuron pathway also play a role in the behavioral effect of these drugs. Although the manipulation on the right, you can see that does not affect the head twitch response. So the interneurons seems to be important for some of the long-term behavioral changes, but not the head twitch response. So this is what I'm thinking about. But to conclude this second part, but also actually our description on our ongoing study, what I show you here is that psilocin act on the 5-HT1A receptor, which actually suppress activity of these SST neurons. But we also think about how the psilocin, the drug, can also directly act on the 2A receptor on the excitatory cell. Again, just highlighting the multiple ways that the drugs can act on the circuit. And then all of these, what does it do? We think it leads to a inhibition, so strong inhibition of the cell body, because the PD cell provide inhibition, suppressing the excitability of the cell body. But interesting, at the same time, when you're shutting down the cell body, which is where the neuron would fire, you're making the dendrites more excitable, both through the SST in the neuron, but also through the direct 2A receptor activation in the dendrites. So all of these, we think, may be a precursors to some of the structural neuroplasticity that we observe. Okay, so I have two more slides. So to conclude, what I show you today is that some evidence that I think psilocybin, even though it induces these long-lasting plasticity, we have found quite interesting and studied quite deeply over the last 5-6 years. What we're quite excited about now is the earliest step on how it might engage the cortical circuits and all these other cell types to mediate some of those effects, including some of the inhibitory neurons, but also some of the non-neuronal cell type. And what I show you today is two studies towards that direction. One is that psilocybin evokes some very cell type specific transcriptional response when you look at the molecular programs. And then secondly, I also show you evidence that psilocybin can reshape inhibitory neuroactivity, by acting on the interneuron, the GABAergic interneuron, like the SST cell and the PV cells. So all of this, led me to think that I think the take-home message here is that psychedelic drug and particularly, or more broadly, psychiatric drug, they really act through receptors, and that's the part we cannot ignore because that's their direct target. But I also think that we should start considering their effect at the level of neural circuits. We should also start thinking about not only on how they're ligands for these receptors, but also how they are also modulators at the circuit level. And this ultimately might be how they elicit many of these interesting behavioral changes and also elicit their therapeutic actions in the brain. And I think by thinking about the drug more as a modulator of neural circuit, it can get some basic fundamental biology insight. For example, thinking about how it contrasts with serotonin signaling, but also maybe as a way to think about how new compounds may be better therapeutic if we start thinking about what it does at the neural circuit level, again, rather than thinking just only at the receptor binding profiles. So with that, I hope you enjoy the talk, and I'm happy to take any questions.
Awesome. Thank you so much, Alex. And of course, if you haven't submitted your questions already, please feel free to put those into the Q&A section of the chat at the bottom, and we can go from with the questions that we do have now. Awesome. All right. All right, so we'll start with our first question. Of the presented research in the presentation, which one or ones do you believe have the greatest potential for human application in the psychedelic roadmap for the future that you're presenting?
I think one piece have some interesting direct relevance. One, but I did go through it quite quickly. Remember how I mentioned about using the rabies virus to map the different circuit pathway that's influenced by the psychedelic? There, one thing that we show is that not only can you figure out which circuits are being modulated, which have relevance because some of those pathways could also be the one that's being modified in the human brain, but we also show in that study, if you manipulate the activity of specific pathway, you can actually tweak the drug-evoked plasticity. What I mean is that the drug normally remodel the circuit in a certain way, but if on top of that, you also manipulate the activity of one pathway, you can selectively change that one pathway in the context of the entire drug-evoked plasticity. So I think that gives some interesting idea. I think that's the first, even though the idea I think is attractive, I think it's really the first evidence to show that you can combine sort of these neuromodulation with psychedelic drug administration. So it really suggests the idea that if you combine, say, drug plus something like TMS, or maybe even drug plus behavioral intervention, where the behavior is seeking to direct the brain activity towards a certain state, that could maybe have some utility in terms of leading to better therapeutic outcome.
Awesome. We got another question coming in. Do you think a psychedelic experience is as complex for the mouse as it is for humans? And is there any proof? The follow-up question is, in relation to that, what is your opinion on the head twitch response being used as an indicator?
Yeah. Well, the first question, I don't know. I mean, I wish I can ask the mouse and see how they feel about this. I had this debate with with other people, whether I mean, if you, nobody will know the answer, but if I were to make a guess, I mean, I do think the mouse is not a tiny human. Their brain is more simplified than us. For example, they're lacking some brain regions like the dorsolateral prefrontal cortex that exists in human, but not in a mouse. Uh, so my guess, again, nobody can prove this is right or wrong, is that I don't know. I don't think they would, I don't think they would be seeing, for example, the mouse god or something, dramatic like this. The second question in terms of head-to-head response as an indicator, I mean, if I have a bingo sheet, right, I would probably win a prize. This always happens in terms of the question being asked. Uh, I view the head-to-head response as a drug development assay. What I mean by that is there are other behavioral assays like that if you're a preclinical researcher. For example, tail suspension or forced swim test. These are assays that exist because we know that they elicit a certain response when you give them a certain drug, let's say SSRI, and they have been very useful for drug development because they have some predictive power. But if you think about the basic behavioral underpinning of it, I don't think anybody would tell you, oh, I really want to understand why the mouse is struggling on a tail suspension. Similarly, I don't think it's worth talking about the biological significance of a hedgehog response. I just think it's a sort of an innate motor response of the mouse. It's very useful as again for drug research, but in terms of what it means for the behavior, I don't want to. I hesitate to go dig deep into that. And in terms of face validity, I definitely don't think it's equivalent to hallucination because we take psychedelic, we don't twitch our head. Only mice do and rabbits do, and to a lot less extent, rats do. Um, but in terms of predictive power, it does predict quite well in terms of whether a compound is hallucination. Overall, yeah, I think it's just good to keep a sort of a even keel mind on what the assay is and what is not.
All right, awesome. We just got a question come in. All right. We got time for some more questions as well.
Yeah.
So if you have any more, anyone else, please feel free to submit them. Do you reckon the impact of psilocybin on glial cells warrants further study, mainly on neuroplasticity and neuronal development?
Yeah, I don't think they're not studied. I think it just we'll see coming up soon. I do talk with quite a lot of colleagues who are studying responses in the non-neuronal cell type. And I think also, yeah, as you just saw, some of the large-scale transcriptomic studies, like the one we did, where the whole thing is unbiased, right? We just study whatever cell that comes out, actually show pretty robust molecular changes in some of these non-neuronal cell, really highlighting that they are also part of the process, which I think then it means makes sense to go in and look at them. And I think one of the probably key part, particularly relevant to some of the stuff we do, yeah, again, is that neuroplasticity, where some of these cell types like microglia or oligodendrocyte, which provides myelation in the brain, could really play a key role because they do support that aspects of the plasticity, which yeah, which no, I think we should study them. Yeah.
Thank you. Now we got a question coming in from Andrew. From your research, do you know if psilocybin is lowering the threshold for PV firing or simply increasing the firing rate?
Yeah, when we record the firing rate is increased, uh, as I show, although why that is, we don't quite know. If you, yeah, again, thinking about the presentation, you notice that I spent quite a lot of time talking about what might be the mechanism for the SST cell, because that's sort of where the reason we focus on that is because overall we're still quite interested in the dendrites, and the SST cells are the one that target dendrites and really excite the dendrite because of their disinhibition. So we didn't actually follow up on the PV cell, why they increase their firing rate. I think one likely possibility is that the PV cell express 2A receptor. So likely the mechanism is the direct binding onto the 2A receptor on the PV cell. So one possible experiment will be to knock out the 2A receptor on the PV cell and see if that's alone sufficient to do that. Um, but I don't know. It's hard to tell without doing an experiment because, yeah, even though I show you, these cell change, increase, decrease, and so forth, at the end of the day, they're also very strongly recurrently connected. They act in terms of the whole circuit. So that's the prediction, but I think we still need to do the experiment to figure it out.
Awesome. This question seems to be somewhat of a follow-up or maybe somewhat
Yeah.
related. This is PV, ALB, and LAMP5 neurons both express the 5-HT2AR receptor. What is the significance of this?
What is the significance to this? Well, I do believe that 2A receptor is the main target of psychedelics, right, including psilocin. So I think it's important to figure out how these cells respond to the drug. The different reasons why the receptor is important. I mean, definitely in terms of you think about the serotonin, it has a lower affinity for some of these 2A receptor than the psychedelic. Uh, so I think as a main target is quite, yeah, interesting to see. I think that's probably the main trigger for what the drugs do in terms of altering cortical circuit activity. Um, yeah, and it's interesting to see that there are multiple entry points. Um, just to say a bit, I don't maybe not exactly what you're asking, but you're looking at it. The other thing that's interesting is that in the excitatory cell, you see it's quite mixed, which makes it a little harder to study because excitatory cell have 2A, but also a little bit of 1A and also some 2C. what's interesting about also some of those PV cell and LAM5 cells, they seem to be quite selective on a 2A. They hardly have any 1A or 2C, if any. That also make it quite interesting because there's a very preferential action on those cell type. Um, yeah, so
Awesome. And I think we got one more question. Might have time for more if any else come in, but what variables do you find to be the most important to address when approaching psychedelic trials in the lab? Or from your experience in the labs, have there been any surprising findings or variables that were discovered?
um, that's an interesting question. I haven't thought about this. Something that's unexpected. Uh, well, I mean, quite a few things are unexpected, I would say. I mean, sometimes we go in with a hypothesis and it kind of turns out the way we predicted. But there are a number of instances where that did not happen. I can tell you that. One is that very first study I told you about in terms of the spine growth. We never expected to find this spine density change to be so persistent. I mean, now it seems like, oh yeah, this was like a study that showed that, but we didn't even kind of set out to do that. The reason that came about was we did the experiment and we were mapping it, I think, up to about 7 days. That was the initial plan. But then the postdoc, Ling Shao, who did the study, 1 month later, basically sent me a message and say, hey, we still have these mice. Should we just image them again? And I'm like, hey, why not? We have them still. So then that's sort of how it came about. We actually never set out to. That's why it's also a little bit, you can also see the way the time point was set up. We measured every 2 days up to 7 days, and then all of a sudden there's a jump to 30 days. But then so that kind of came serendipitously. Like we never actually even thought that it was sustained for so long. The other thing that was quite interesting was the rabies study that we did. It was also have some surprises. The way we built the study, again, is digging into the weeds, but we really were inspired by a rabies tracing study of cocaine that was done by Rob Malenka at Stanford. And in that study, Rob and his colleague Kevin Baer showed that with a dose of cocaine, and then you trace the rabies, you find a few pathways that's being strengthened or weakened. So we thought when we go in the rabies study, we would also find a few pathways being modified by the psychedelics. That was a prediction, but when we actually do it, we find it's a fairly brain-wide change. We find many pathways that's being modified by the drug that's quite different from what was reported previously for cocaine. Yeah, so I think it was in science that happened quite often. You know, you go in with a particular prediction. I think you do want to go in with a prediction because you want to see whether it has some framework to start with, but then whether it matches it or whether you find some counterintuitive result. I think that's where the fun comes in. And you have to, have an open mind because, yeah, once we figure out that's what happened with the rabies result, then we basically change our thinking and later on develop our experiments towards those results. Um, but yeah, I would say those are a couple of the sort of unexpected turns in our studies.
All right, awesome. Well, thank you so much for your time. Thank everyone for their questions. Of course, everyone, once again, thank you to Alice Kwan for sharing their expertise and their time on day 3 of PsychedelX.